Starting recording. Here we are now recording. Side note. Problem with last time is oh my goodness, stranger among us. I uh, Last time there was a they let their license expire on this. It's not the fault of that. This happens all the time. But what happened is while they were trying to get through the paperwork for this kind of stuff, that's why we were unable to record last time. So I'm going to have to do a bit of it over again. And I'm going to do it from the other PowerPoint, which I got a chance to load and look at. It's similar. It's arranged a little bit differently than the Nestor PowerPoint, but it accomplishes the same thing. So I'll do maybe 20, 25 minutes of that and then try to get a little review in as I promised, because the person I promised it to has made it to here today. Another rare sighting. What?
Do we have a picture of the sword arch?
I have a video of the sword arch. I'm sorry. I'm figuring out how to do that. Okay, but if you want to come by later, you can watch it on my phone. It's very cool. I'll tell you what I'll do is I'll get in touch with my daughter-in-law, because I have no idea where she put. She's got like 48,000 pictures on Facebook, so I have no idea where she put that stuff. Which is the only reason I have Facebook, is to look at pictures of my kids and grandkids and any other kids that might be part of the problem. Moving on. So here, you'll find it here, the Cowan Microbial and Human Interactions. This one we're doing. And then the next, and basically this accomplishes, it's effectively these two chapters are our next exam. Okay. So that's where they're going to be, and the antimicrobials. And we're going to have this. I'll add the Nestor PowerPoint and some outline materials for antibiotics, and everything should be all accomplished with regard to that. So again, for you late arrivals. No, sorry, not it. Because we didn't have the record function properly, I figured I'd do a little bit of introductory stuff, and maybe in about 20 minutes or so, then hop on with some review for the exam whenever day you're planning to take it, which is fine. Like I said, I'll be here. We'll be here Friday, and we'll be here on the Monday when you get back. Can't wait to see none of you. And moving on. That's all right. I'll be in lab today. I'll be watching a soccer match. So you guys are doing a lab exam. So try not to bother me. I brought in my iPad for that. FA Cup, round of 16. I'm worried about a letdown. But I digress. Okay, here I am. All right. So it's interesting. They sort of hybridized a couple of chapters. They all use the same materials because that's what they do. And so it's really a little bit about how these things make us sick and why they're so abundant and why they're everywhere and how they spread. And really, that's what this is all about. When we ended, for those that were here last time, we ended with virulence. What we're looking at are different factors why things are nastier than others. I mean, to me, maybe the most important factor is your overall physical health. I think your best gift to yourself is trying to keep as healthy as possible, whatever that entails, whether it's diet, whether it's exercise, whatever you're doing, allowing yourself to heal if you get sick for whatever reason. Those are all the kinds of things that really provide you with a certain degree of ability to survive. I mean, I take it from somebody other than being around, but have treated a lot of patients over not in the last 20 years, but in the prior 20 years before that. It's an accumulation of battles. And you get to a certain point when you have too many battles in a row, it's hard to survive. And that's what we're seeing now as folks get older. I'm speaking about my mother-in-law. I mean, she hasn't got another battle left in her. One more, that's going to be it. I mean, she's 96. God bless her. You know, it's okay. But the reality is you can see it every time there's a challenge. So, and it's really your own physical ability and physical wellness that helps it. That's my little spiel on that. So, and so they talk about the microbiome, all the microbes on it. This was interesting And because we're discovering they're more and more important. It's a certain amount of us getting used to them, being constantly exposed to them, but problems if they get where they shouldn't be. And they're in a lot more places now than we thought. Okay. Colonize us for the long term, don't cause diseases. We call them resident or resident microbes. And in the old days, we call them normal flora. Okay. If somehow, not just these, but any can get past our host defenses, enter tissues and multiply. You're probably in the process or started, or depending on who's, I don't know what Dr. Robbins is doing. Are you doing some immunology now with him? Yeah, it's about right. I know how I would sort of schedule it for when I would do AP2. And this is the idea. There's a series of boundaries, and and part of that general health thing is maintaining those boundaries. And when these get through, or even ones that are there ordinarily, the resident ones, the normal ones, if your boundaries are weakened because of some what we fondly call comorbidities today, then that changes. So deviation from health is when they disrupt some effects sufficiently for us to demonstrate, and again, signs or symptoms. The whole idea, a symptom can be any kind of a complaint. It's subjective. It's you feeling it. A sign, observable and measurable. Whether it's a temperature, whether it's a blood test, whether it's a physical observation that you make. If somebody's got a piece of bone sticking out, obviously they're injured. It's not rocket science. Okay. Infectious diseases are caused by microorganisms. That's kind of our aspect of it. The microbiome project's been around for a long time. We have to understand, how they affect us, how they're altered. We use a lot of genomic tools, being able to sequence and understand it. I think that's of interest. It's not terribly important for our course, but you can see we've got a lot of genes that encode for proteins, but microbes. If you want to take of all the microbes that are on us, they have millions of them. And we find them in locations that we didn't used to see them. Look alone at how many in a gram of, it's not a whole lot of, as we finally get a poo poo. Because I don't want to say that other word that's associated with it. Okay. So imagine how many viruses you're contending with on a regular basis. So we all harbor. We do. I mean, it's the notion that, oh my God, stay away from that person, or it's really not realistic. I think it's more about you, unless someone's so obviously ill and contagious that they would provide a health problem. You just don't know where it's coming from. Because as we talked about it, for those that weren't here last time, asymptomatic carriers, people who can bring the disease to you when they don't even know they have it. So there's a lot of, particularly gastrointestinally, these are things that they're looking at. So we knew skin, mucous membranes, linings, upper respiratory, GI, particularly the mouth, I think as much as any, external portions of the genitalia, urethra, vaginally, all these external exposed surfaces. Okay, now we're starting to look at the brain and the bloodstream. We're actually seeing evidence transiently of microbes. Frequently when you eat, certainly when you go and get your teeth cleaned, you have what's called a transient bacteremia. There's nothing unusual about that or problematic unless you have a pre-existing condition like a damaged heart valve. And sometimes folks have to have antibiotic prophylaxis. At least they did years ago. It probably has changed a little bit. Where they had, if they were going to get scaling and cleaning, they had to take prophylactic or preventative antibiotics. It's just the way it was. Now, it may have changed significantly with whatever the thing is, and the best person to talk to be your dentist about that. So as you can see this, now we're starting to even begin to see things in the lower respiratory tract, within the bladder, the urine, which was always thought to be sterile, breast and breast milk, amniotic fluid, okay, etc. So there's a lot of different things that are part of it. Okay, we have a lot of different range of environmental areas based on all of these factors, nutrients, pH, etc. There's a certain amount of competition, and we talked a little bit about that before, because the good thing about the normal microbes is they're fighting for not so much you, but fighting the bad guys for spaces where they could lodge. And that's useful to us. and they create an adverse environment frequently by their waste products, which act as sort of enhances the level of competition, typically to our benefit. If they're there and they're making stuff that the other guy can't tolerate, it's a good thing. Okay. So when they talk about, and then we looked at those definitions between mutualism and commensalism, and I never really could distinguish it because neither side's really being hurt. So that's a good relationship that's there. Other than, as you'll see here, a truly pathogenic relationship where one party flourishes and the other party is damaged. And that's typically what happens when we're invaded by something. Okay. So that's part of it. Different things that we get pretty obvious. Okay. Infancy or prematurity, certainly as we get older, we're much more predisposed to problems. You can't expand your lungs as much, your bones are more brittle, your blood vessels are not as nice and springy or elastic. Those are all signs of a unit I hate doing in AP2 or in my advanced physiology, the aging or senescence unit. Don't ask me why. I don't like it. Okay. That's why I asked my wife this very same thing yesterday. What did you do, marry some old guy? She's considerably younger. Did I tell you where we met? Did I? Yeah, I told you. She was my student. Don't start. It was 20 years ago. Okay. There was only 7 years difference in our ages. It's not like I'm robbing the cradle. It's not like a May December marriage. It's more like an October December marriage. We're sufficiently close together. I'm deeply in touch with my dirty old man. Okay. Don't start. Do not repeat. Don't repeat this. Moving on. I saw that. I saw that look. So different things. Obviously, immunity acquired, immunological deficiency. They always talk about AIDS, but it's not certainly not the only one. Different types of surgical intervention, transplants, because you almost always have to be on immunosuppressants, underlying diseases, immunosuppressants, stressors. Pregnancy is a major stressor, concomitant infections. That's that idea of primary versus secondary infection. If you have an infection, it weakens your defenses. They're occupied with one, and it may either damage the tissue or predispose you to yet another kind of an infection. Okay, so this is interesting. Okay, the uterus and its contents were thought to be sterile during embryonic and fetal development. Okay, but even we see this by way of the placenta. So maybe it's biologically a way to begin to colonize early on, because colonization starts immediately at the birth process. And as you might remember, we talked about it on Monday for those that were here. It's a little different versus cesarean birth versus vaginal birth. The type of colonization. What that means long term, I don't know. Okay, so vaginally, lactobacillus, necessary enzymes to digest milk. Lactobacillus is such an important player, and that's really the major player vaginally. Other species from different kinds of other disorders. Human milk contains about 600 species of bacteria and sugars the baby can't digest, but are facilitated by healthy gut bacteria. I mean, understand, effectively, we're all born lactose deficient. That's why it's very, standard cow's milk is problematic. Okay, at the very least. And it takes a certain amount of time to make that transition to the whole milk products from formula. And if you're like my kids, they, between the soy milk and the pre-digested milks and all that type of stuff, it was, which all smell. So all of these types of things contribute. Milk has a microbiome of its own, caregivers, other people, the environment itself, all of those types of things. So the images are pretty good. I'm not worried about that. So pathogenicity levels and virulence is really maybe the most important subset of what we learn about in this particular section. what makes things nasty, how it gets to the point where it can cause the disease. It's interesting. Our discussions are more going to be about the causative agent. There's a lot of polymicrobial infections. A lot of times it's a question of trying to treat the primary culprit more so, and sometimes you have to use multiple antibiotics. I saw a ton of these in diabetic patients, a ton. We treated what are called diabetic peripheral ulcerations. Their skin breaks down. They get lots of infections. You'll get sick and tired of them if you're going to be in a clinical field. Okay, they're a bear to treat because a lot of times there's combinations of gram positives and gram negatives, all those types of things. How they enter and how they exit, not always the same. Okay, what the infectious dose is. Again, that's that ID50 we talked about a little bit. How they cause damage. Right back to something we learned early on, endo versus exotoxins, the virulence factor. I don't know if we have to diagram anything. And so, again, what's a pathogen? Relationship is parasitic by definition, results in infection and disease. So, and then we talked about this a little bit. True pathogens capable of causing disease in healthy persons with normal immunological defenses versus opportunistics, which we see in folks who have already what we call a compromised host. Today, they already in the COVID, the comorbidities and stuff, same old stuff. Okay. And so virulence factors that are there, the severity of the disease is effectively what virulence is. So we look at a variety of factors that are virulence factors. And that was something we started to look at the very end of the presentation on Monday, the degree of pathogenicity. So virulence doesn't mean virus, once again, even though we utilize that word, at least at present, to describe it. Its ability to create an infection, in other words, establish itself in a host, and the nature of the damage, and any characteristic or aspect, structure of the microbe that contributes to that, its ability to cause damage, how reactive you are to it, how nasty. To me, it's like a nastiness measure. Those are virulence factors. Here's the idea. There's typically more than one contribution. Usually there's a primary culprit, I would say that, or an underlying disease or condition. I mean, it's simple. You all did the skin unit. When you burn yourself, there's two major things that impact you: fluid and electrolyte loss, and the likelihood of infection, because burnt skin is comfort food. No other way to say it. For a variety of bacteria, particularly the aforementioned Pseudomonas aeruginosa. Okay. Influenza, classic example. Viral infections are typically not fatal in nature. What happens is they cause a secondary infection with bacteria, particularly bacterial pneumonia in this case. We talk about viral infections of the lung. And what happens, that's a lot more damaging. Okay, because of the way bacteria work as opposed to the way viruses work. Several types of staph and strep. Okay, so here you can even get, and here's more axilla, which is actually a normal flora. So staph and strep, you all should know, are gram-positive cocci, one in clusters, one in chains, right? Remember that lab, that type of stuff? I know that. And Moraxella is actually in the Neisseria family, kind of a distant relative, so it's a gram-negative Diplococcus. And then we look at the different portals of entry, how it gets in, how it gets out, whether it comes from the outside or whether it's something that's already inside of you or on you, as it were, whether it's part of you. So that's where we begin to see some of these opportunistic infections. Okay. And so I think that's all part of it. So, and I mean, it's a little interesting here, adapted to a specific portal that provides the most favorable way for it to grow and spread. So most of the time it's this way. Every so often you get the oddball that I was talking about. We mentioned a bit at the end about prions. I just sort of amplify what I sent you. And really, the prions are typically thought to be ingestion, but if you get direct impact from a contaminated transfusion, you can obtain the disease that way. So if certain pathogens using the wrong portal would not be infectious. I mean, again, you could get influenza virus on your fingers. You're not going to get sick, but you touch your face so often, you're going to inhale it. So what's the answer? Hand washing, hand washing, hand washing, hand washing. Okay. Virus contacts the skin, no infection. That's the example. Some more than one portal. Tuberculosis can enter respiratorily, also GI. It is by far and away much more common respiratorily. But what's the major underlying factor for tuberculosis? Overcrowding. So you're much more likely to come in contact. Someone's coughing with tuberculosis and they cough constantly. It's going to get on the food. It just is. Strep and staph enter through the skin, urogenitally, respiratory, because they're all over the place. All right. So that's 20 minutes, and I'm going to spend the rest of the time doing the review. So allow me to exiting. See, I promised you I was going to do this. I'm going to do a separate recording for